Welcome back to the channel, everyone. Today we are going to be talking about centrifugation techniques. Now, centrifugation is one of the most widely used techniques in all of biochemistry and molecular biology. And what it does is that it simply separates components of a mixture based on size and density using centrifugal force. So this is a laboratory centrifuge. It should be noted that centrifugation is also used outside the lab in industries that separate other kinds of mixture, such as the petroleum industry. Now, the principle of centrifugation is quite simple. When a sample is spun at high speed, the centrifugal force pushes the heavier particles outwards, forming pellets, while the lighter particles remain suspended as the supernatant. Now, the rate of this sedimentation is dependent on the size and the density of the particles as well as the viscosity of the medium. So, sucrose, for instance, is more viscous than water. So, the rate at which a particle would travel through sucrose to the bottom of a tube would definitely be different from how fast it will travel through water. Now let's look at the components of a centrifuge. The centrifugal spinning machine um, consists of all the moving parts. The rotor, which rotates, this is where the tubes are fixed into, and then there is an electric motor that actually powers the rotor through the drive shaft. There are also tubes wherein the samples to be separated are kept, and tube holders which hold the tubes, and a chamber where all of this is housed. There is a latch and a lid for this to be covered, and there is also a control panel. Now, on the control panel, you can control the speed at which the centrifuge will operate, as well as the temperature in some systems and the time for the spinning to occur. So all of that can be preset during the centrifugation process. Let's look at the types of centrifugation. Start off with differential centrifugation, which separates particles by size and density through successive spins at increasing speeds. So if you have a mixture like this, after the first round of centrifugation at a certain speed, these heavier green particles will be the first to sediment. So thereafter, after they've been separated, the supernatant is taken into another tube and is centrifuged again, this time with a higher speed. And then the biggest particles, which is now the red particles, will be the ones to sediment to the bottom. Thereafter, supernatant is taken and the process repeats like that. Now, this process is used in the fractionation of cells. So this is how differential centrifugation is used in fractionation of cells. So you have the cell lysate, which is what you get after homogenizing a cell. When you centrifuge at 1000 G for 10 minutes, the whole cells, the nuclei, and the cytoskeleton are the first to be separated. And thereafter, the supernatant is taken and spun at 2000 G at 20,000 G for 20 minutes for the mitochondria, lysosome, and peroxisome to be separated, and the process goes on like that. Now, each of these pellets that have been separated can be reconstituted or taken elsewhere to study further, and this is how scientists study different components of the cell. They separate them using differential centrifugation first. Next, we look at density gradient centrifugation. In density gradient centrifugation, the particles migrate through a medium with a density gradient, such as sucrose, until they reach a position where their density equals that of the surrounding medium. So sucrose has an increasing density gradient. So the bottom of a tube containing sucrose is more dense than that of the density at the top portion of the same tube. So when organelle fraction are placed at the top after centrifugation, the different organelles would migrate to the part of the tube where their density matches that of sucrose in that tube. Now, this method can also be used in separating blood sample. Here, blood sample is added to fecal. Now, fecal is just a brand name for synthetic branched polysaccharide made from sucrose and epichlorohydrin. So, it is added to the blood sample, and after centrifugation, the blood would separate like this with plasma um, on the upper part of the tube, white blood cells and red blood cells at the bottom. Now, this method of centrifugation, the density gradient centrifugation, is more precise than differential centrifugation and has two main forms, the rate zonal and the isopycnic centrifugation. And we're going to look at both of them in a little more detail now. In the rate zonal centrifugation, the particles are separated by size and shape within a preformed density gradient, and it is time sensitive. So larger particles sediment faster before they even reach equilibrium. And in this kind of centrifugation, the sucrose gradient is stabilizing and shallow. So it's just 5 to 20% sucrose that is used. Now, after centrifugation, the fast sedimenting components migrate to the bottom, and then the slow sedimenting components remain towards the top. In isopycnic centrifugation, particles separate purely by density, independent of time. So each particle settles at a position where its own density equals that of the medium. This method is ideal for purifying acids or organelles. Also, in isopycnic centrifugation, there is a steep sucrose gradient, so it's between 20 to 70. So, this is a much more concentrated sucrose mixture. And after centrifugation, um, particles with high buoyant density migrate to the bottom, and those with low buoyant density remain at the top. Next, we're going to look at analytical ultracentrifugation, which is used for studying macromolecules like proteins and nucleic acids in solution. It provides data on molecular weight, shape, and interactions by analyzing sedimentation velocity or equilibrium. Now, this kind of centrifugation combines an optic system which helps to give the data on molecular weight, shape, and interaction. It is basically like combining centrifugation and spectrophotometry to monitor in real time the absorbance of the particles being separated. So it is very complicated. Let's look at some applications of centrifugation. One, it is used in isolating cells and organelles from tissues, as we have seen. It's used in purifying macromolecules such as proteins and nucleic acids. It's used in separating lipoproteins in clinical diagnostics. It's also used in clarifying solutions or removing debris before analysis. In summary, centrifugation uses centrifugal force to separate components based on density and size. Differential centrifugation is ideal for basic separation, while density gradient offers much higher precision. And analytical centrifugation provides advanced applications for research and large scale purification, especially in the enzyme industry. So thank you so much for watching. Please remember to give this video a thumbs up, subscribe if you haven't already, and let me know if you have any questions in the comment section. I will see you next week. Bye.